And so this is a picture of a hippocampus, mouse hippocampus in this case. And all the studies that I will describe from this point on came from a paper in Nature 2007 by Levitt et al. And this work was done at Harvard University in collaboration with scientists at MIT. And they call this technique Brainbow, which is brain plus rainbow. And it gave staining in more than 100 different colors, as you will see in the next few slides. So the inset in the previous diagram is enlarged here, and these are higher magnification of hippocampal cells. A hippocampus is where memory and spatial navigation are located or controlled, and the larger cells, which are highly branched, are the neurons, and the smaller cells are the glial cells. And the confetti-like stuff are various remnants of signals. How was this color circuit staining achieved? And Levitt et al. Used 3 or 4 different color fluorescent protein genes that were transferred into the mouse nervous system, and combination of these genes expression results in the colored proteins that give the variety of colors seen in the different neuronal cells in the previous figures. Now, what does incompatible lock site means? Well, first, lock sites were from phage P1, and they are roughly, as I remember, 34 base pair sites where an enzyme called Cre carries out site-specific recombination between 2 LOX sites. So the 2 different LOX sites used in Brainbow 1.0, one was the original LOX site, LOX P site, and then the other one is a minor variation of LOX P such that it can still be recognized by Cre and used for recombination, but the canonical LOX site and this variant cannot recombine with each other. Okay, and so we will go to this slide, Brainbow 1.1, in order to understand exactly what's happening. So in Brainbow 1.1, there are 4 different fluorescent protein genes with a brain-specific promoter at the left. And next to the promoter are 3 different incompatible LOX sites. And then in between the fluorescent protein genes are a single LOX site. So between orange and red fluorescent protein gene is LOX site number 1, and in between red and yellow fluorescent protein gene is LOX site 2, and between yellow and blue is the LOX site 3. And then there is a poly A tail signal, etc. Now, in the absence of Cre enzyme, whatever protein gene is closest to the promoter is translated into a protein. So cells without Cre express the orange fluorescent protein as shown in the bottom left diagram. Once Cre is brought in trans, recombinations happen, and because all the LOX sites are in direct orientation, recombination between them causes deletions. So when recombination 1 happens, the orange protein gene is deleted. If recombination 2 happens, then orange and the red fluorescent protein gene is deleted, and recombination 3 leads to deletion of orange, red, and yellow fluorescent protein genes. And so depending upon which of these 3 different recombinations happen, either the red or the yellow or the blue fluorescent protein gene is brought closest to the promoter, and you end up getting cells that are red, yellow, or blue. And so you end up with a mixture of 4 different color fluorescent protein being produced and 4 different color cells. Now in Brainbow 2.1 shown here, there were 4 different fluorescent protein genes. The green was closest to the promoter, then yellow, then red, then blue. And a single kind of lock site was used, but there were 4 copies of it. So the leftmost copy and the rightmost copy are in inverted orientation, and the 2 middle copies are also in opposite orientation. And so many different kinds of recombinations can happen between them. For example, locks site 1 and 3 are in direct orientation, and locks 2 and locks 4 sites are in direct orientation also, so they cause deletions if Cree recombines them. In contrast, locks 1 and locks 2 sites are in inverted orientation, and locks 3 and locks 4 sites are also in inverted orientation. So recombinations in these 2 cases leads to inversions. And so depending upon which of the 5 recombinations shown here, 1, 2, 3, 4, 5, you can get cells that are expressing green if there is no recombination, as shown at the bottom left. And if different recombinations happen, you get red, blue, and yellow cells. So you end up getting a mixture of 4 different kinds of cells. And all these studies were done with cells in culture. Now, when they went to the mouse brain cells, they got more than 100 different colors. So the question is why? And that's explained here in this diagram. And what happened is somewhat similar to how we get millions of different colors in a color television. Color television has many, many pixels, and each pixel is one of 3 colors. But because there is such multiplexity, we end up getting millions of different color combinations, and the pixels are so tightly packed that we don't see individual pixels. We see groups of them, and the whole picture has many, many different colors. So this diagram shows 3 different color fluorescent protein genes: blue, green, and red. And there are 3 copies of each. So depending upon which combination of these 3 color genes are expressed in these 3 copies, you end up getting 10 different colors from blue to gray. And Livet et al. Estimate that they had as many as 10 of these 4 protein gene cassettes inserted in the mouse brain cells, and that's why they got more than 100 different colors. So in conclusion, using modern genetics technology for the first time, people are beginning to imagine what seemed to be an impossible goal, because you can do so many different color staining that you can follow axons in the tangle they exist over long distances. So the ultimate goal is to push the staining techniques far enough to be able to create a complete connectome map of the brain cells. Now, given that there are billions of neurons making trillions of connections, my guess is it will take a while before the technology is advanced enough.